homology of the nucleotide and amino acid sequences of enzyme 1 and 5 in pathogens pathway of isoprenoid biosynthesis. And you can tell biology is not necessarily my first area, but thank you very much, Haley, for what you present.
Thank you, Dr. Myers. So now you may be wondering how I came upon this topic as the focus of my research. What sparked my interest in this was the 2-week program I participated in this summer called BioQuest Academy, hosted by the Seattle Biomedical Research Institute. Here, in a laboratory setting, I was introduced to themes of scientific research involving the infectious diseases, tuberculosis, HIV, and malaria. I was also introduced to broader themes of global health. Additionally, what motivated my motivation to pursue research in the field of global health was my trip to Haiti last year, where I witnessed firsthand huge populations of people living in the poorest health conditions imaginable. Infectious diseases is not just a local or national problem, but it's actually a global health problem. Infectious diseases combined kill more people worldwide than any other killer. And although some drugs may exist to inhibit these infectious diseases, it is important to continue to pursue scientific research because of the way pathogens rapidly gain drug resistance. Initially, I wanted to focus my research on the disease of malaria. Malaria is caused by the protozoan parasite of the phylum Apicomplexa and the genus Plasmodium. It is a significant problem because it kills approximately 1 million people each year, most of these being children under the age of 5 years old. Next, I wanted to expand my research to include other infectious diseases. Most of these also pose great global health challenges. Here you can see the visuals on my slide. So from here, I developed the goal of my research being to identify a possible drug target within the Plasmodium parasites and then expand upon this to see if the same drug target could be viable in other pathogens. Although drugs work in many different ways, one way in which they function is by targeting a specific pathway of the pathogen and inhibiting a vital enzyme involved in this pathway. When I was starting my initial research on the Plasmodium parasite, and especially the cellular makeup of the organism, I discovered their unique organelle called the apicoplast, and identified it as a potential promising drug target. The apicoplast is a non-photosynthetic plastid present only in protozoans of the phylum Apicomplexa. The apicoplast is vital because it houses the non-mevalonate pathway of isoprenoid biosynthesis. Isoprenoid biosynthesis is a necessary metabolic pathway that produces isoprenoid molecules. This group of compounds carries out a diverse group of functions, as you can see on the slide. It is completed by one of two ways, either the mevalonate pathway or the non-mevalonate pathway. The non-mevalonate pathway is found in most bacteria plants and apicomplexan protozoans. It is a multi-step process that involves the use of many different enzymes. However, after reading multiple studies, I became mostly interested in the enzyme 1-deoxy-D-xylulose 5-phosphate reductoisomerase. And this is a potential drug target because it is not found in human cells. Human cells use the mevalonate pathway of isoprenoid biosynthesis. This does not include the use of the DXR enzyme. From here, after identifying this enzyme and becoming interested in the presence of the enzyme in multiple different pathogens, I developed my research question. How conserved are the nucleotide and amino acid sequences of the DXR enzyme within the genomes of multiple parasites in the context of the development of a universal inhibitor? Initially, because I was interested in the malaria parasite, I chose 4 different species of the genus Plasmodium to analyze. And then, because of my interest in the apicoplast, I chose 2 more apicomplexan pathogens to look at. And finally, to expand the breadth of my investigation, I chose to add 4 more pathogens, all of these being prokaryotic, and also posing great global health challenges. One way in which I was limited in this step of my investigation is that I was restricted to only choosing pathogens whose genomes have been published by the National Center for Bioinformatic Information's Genomic Database, or NCBI. For example, this can be seen in the fact that although I looked at 4 Plasmodium parasites, only one, Plasmodium falciparum, actually causes malaria in humans. This was a major limitation. Another way in which I was limited is that because the NCBI did not have the sequence for the catalytic domain available, I was restricted to only analyzing the C-terminus domain of the enzyme. The C-terminus domain, seen here, although not directly involved in substrate binding, is indirectly involved because of its close proximity to the catalytic domain, seen here in green. in looking at the potential broader implications of my research, I developed my hypothesis. If the amino acid sequences show high homology, meaning the DXR enzymes of the various organisms are similar, then there exists a better potential to develop an inhibitor capable of being effective against a wide range of pathogens worldwide. My investigation is made up of a 5-part procedure based in bioinformatics. The first step I took was to obtain these nucleotide sequences, again looking at NCBI. Here shows the list of available protozoan genomes, including the Plasmodium species. From here, I navigated to search for my DXR enzyme and repeated this process for each of my 10 organisms. The next step I took was to use the ClusLW multiple sequence alignment tool to align these 10 nucleotide sequences. ClusLW identifies areas of homology between multiple sequences to produce alignment scores. One alignment score is produced when 2 sequences are aligned. So, by inputting my 10 different sequences, I obtained 45 unique alignment scores. When aligning nucleotide sequences, ClusLW takes into account the number of identical nucleotides. And when analyzing protein sequences, it not only takes into account the number of identical amino acids, but also the similarity of these amino acids. Scores between 0 and 100 are produced, with scores approaching 100, approaching 100% homology. Here are the results from this step of my investigation. Here I have color-coded my matrix so that you can see the widespread moderate homology, seen mostly in the green squares. There's also higher homology shown when looking at the Plasmodium sequences aligned with each other. Another interesting thing to point out is that the tuberculosis sequence, when analyzed against the Plasmodium sequences, did not yield as high of alignment scores, possibly because of some sort of evolutionary divergence. The next step I took was to translate these nucleotide sequences using the XPASI translate tool. And then, in order to ensure that my newly translated protein sequence is still coded for the DXRC terminus, I used the NCBI's BLAST tool to verify that my sequences align with known DXR proteins. And finally, I used the ClusLW tool again to align my amino acid sequences. Here is the resulting matrix from this alignment. Again, you can see the higher homology when aligning Plasmodium with each other, as well as the moderate homology overall. Another interesting thing is that overall, the alignment scores of the amino acid alignments were much greater than the nucleotide alignment scores. This is because it is possible for multiple different nucleotide codons to code for the same amino acid. So, although the nucleotide sequence may be different, it is possible that it codes for the same amino acid sequence. Here I chose to graphically represent my data with bar graphs. These 4 graphs show the 4 species who produced highest alignment scores to each other. These are the 4 Plasmodium species, and this makes sense as they all have the same genus. Next, these are the 2 apicomplexan pathogens that were not malaria-causing pathogens, B. bovis and T. parva. Here you can see that they also exhibit moderate homology overall, however relatively higher homology to each other. This is because they are both of the order Pyroplasmida. And finally, I looked at my prokaryotes. Although because prokaryotes as a group are generally more diverse than the apicomplexan protozoans I looked at, I found that each prokaryote identified with one other to a moderately high extent. For example, tuberculosis and C. diphtheria produced a relatively high alignment score. This is because they're both of the order Gamma proteobacteria. On the other hand, I found that B. cholerae and S. typhi produced a relatively high alignment score to each other. This is because they are both of the prokaryotic order Actinomycetales. Here, before proceeding on to my next visuals, it is necessary to review over the significance of the chemical properties of amino acids. When aligning 2 sequences, if one amino acid is substituted for another, however the new amino acid exhibits similar chemical properties to the initial amino acid, the folding of the protein and the enzymatic activity will not be as greatly affected. In addition, it is necessary to clarify over what the difference between a very similar amino acid and a somewhat similar amino acid is. Here is an example of a group of very similar amino acids. They all share the specific chemical property of being aliphatic. Here is an example of somewhat similar amino acids. These, although share the same property being polar, there exists some key chemical differences. Here is a visual produced by ClusLW. ClusLW, in addition, identifies areas where the amino acids are identical, very similar, or somewhat similar. I've color-coded them so that you can see the relative moderate homology. The next step I took was to repeat this process. However, this time only looking at the Plasmodium pathogens. This time I found a much higher degree of homology. So here in the colored sequence, in the colored region, you can see a sequence of 230 amino acids where nearly all of them are identical. In looking at this sequence, I found that 77.8% of the sequence was made up of identical amino acids. And when I expanded my scope to include very similar amino acids, I found that this percent identity score increased to 92.2%. It's important to look at this percent identity with both of these scores because as these proteins are made up of amino acids with similar chemical properties, they will fold in similar fashions, and so it may be possible to develop a drug capable of inhibiting the two different enzymes in similar ways. In conclusion, the C-terminus region of the DXR enzyme is moderately conserved throughout the genomes of all 10 of my organisms, with higher conservation being shown in the genomes of organisms who most closely share evolutionary history, such as these. Now, what's the next step? The next step would be to look into how we might inhibit this enzyme. Although initially I chose the DXR enzyme after reading only a few published studies, afterwards I found current drugs being investigated as DXR inhibitors. This confirmed my research. For example, on the left you can see there is a drug called phosphate. This is being researched as an anti-malarial and is currently in the process of human clinical trials in Gabon and Thailand. In addition, there is a similar group of novel compounds called phosphonohydroxamic acids. This is being expanded as a DXR inhibitor to inhibit prokaryotes such as E. coli. So, although the scope of my research only included a tiny fraction of the total process of drug development, I was introduced to the preliminary steps of pharmaceutical research. For my project, I gained a greater understanding of what it takes to tackle these huge global health issues that affect the poorest and most vulnerable populations of the world. Acknowledgements, my bibliography. Are there any questions? The question was how big is the protein overall and what percent is the C-terminus domain. I found that the nucleotide sequence of just the C-terminus was around 1,000 nucleotides long. And so although I do not know for sure, I would imagine that this makes up approximately a quarter of it after looking at the diagram. However, I'm not completely certain. Yeah, so the question was regarding my experience at Seattle Biomedical and to what extent I did the work there or versus by myself. So when I was at BioQuest Academy, I did a kind of similar research project involving malaria and the possible development of a vaccine looking at specific proteins being expressed on the erythrocytes being invaded by pathogens. Here I was introduced to the bioinformatics tools that I used, the QuestLW, as well as I was kind of taught how to navigate the NCBI. However, I identified the DXR enzyme independently and carried out my research mainly independently. The question was, what are my career plans? Right now, my career goal is to become a pediatric infectious disease specialist.
Judges, more questions?
Will you have opportunity to do any follow-up research at this point?
At this point, I don't see myself doing follow-up research. However, in the future in college, I would like to continue to study bioinformatics as well as molecular biology, involving infectious diseases such as malaria.
Will you be able to share your findings with someone to give a follow-up?
The question was, will I be able to share my findings?
Will you share your findings?
Hopefully, yes. Yes.
Just out of curiosity, if following the development of successful
So the question was, if I were to follow up on some of these inhibitors that I mentioned, kind of how long would they last and how would I study the emerging drug resistance? So currently, it is in human clinical trials, and I would imagine that it would last for years, however, it lasts longer in certain regions of the world. For example, there is a current anti-malarial, the most common one, quinine, and it has existed since malaria existed. However, currently in certain regions of the world, there is emerging drug resistance, so I would imagine it would last for different amounts of time in Southeast Asia versus Africa versus Central America, and then from there I would attack it with a multi-drug approach.
What effect would you expect the inhibition of DXR to have on the non-pathogenic biology in humans? So would it have any negative effect if I were to inhibit the DXR that's operating around?
Oh, so the question was, what would the effect of the DXR inhibitors if I were to take it without the pathogens? I would imagine it wouldn't directly affect the human cells because it doesn't contain DXR. And I would also not expect it to have too much of an adverse effect to the natural symbiotic bacteria in our system, just because of the way other anti-malarials and such don't have much of an adverse effect. But again, because it is an antimicrobial, it could have some sort of adverse effect, so you would not want to take it for extended periods of time. Yeah.
How would a DXR enzyme inhibitor work differently from like quinine, like the drug that you mentioned?
Excellent question. So the question was, how would these DXR inhibitors work differently from the common antimalarials being used? So the common antimalarials being used, such as quinine and chloroquine, they cause the buildup of the cytotoxic hemigroup that is produced while the pathogen is kind of digesting the hemoglobin inside of the red blood cell. And so how those work is by causing the pathogen to almost suffocate on its own waste. And this would instead just target an enzymatic pathway.